friends, welcome you all my YouTube channel Aky Biotech. Friends, this video is very important and today we will discuss about the novel coronavirus. We can also call it COVID-19, and we will be discuss in the two part. In the part 1 video, I will discuss about the structure of the novel coronavirus COVID-19, and in part 2 I will discuss the detail of the molecular structure of the novel coronavirus. Friends, before starting this video, if you are new at my channel, please subscribe the channel. If you have any doubt, please do comment on the comment box and like and share the video.
Starting with the coronavirus, that is SARS coronavirus 2 COVID-19. About the structure, it has created pandemic all over the world. It started from Wuhan, that was the epicenter, and day by day it has spread to all the countries. Mainly the countries which are affected are United States, Italy, Spain, as well as India. SARS, that is severe acute respiratory syndrome. It is called as COVID-19 because it is a novel coronavirus and it started in the year 2019. Coronaviruses, a little information about them. These are the enveloped, positive sense, single-stranded RNA viruses that belong to the subfamily of these viruses is Coronaviridae and family is Coronavidae. Looking into the structure, as you can see in the figure, this is the spike glycoprotein. Next, in between the envelope, that is the nucleocapsid. Then comes the membrane protein, the outer one. Next is envelope, and the inner coil structure, as you can say, that is positive single-stranded RNA. This is about the 3D structure. COVID-19, it's a beta coronavirus and it was discovered in Wuhan city, Hubei province in China in December 2019. Coming to the viruses, these are the enveloped. Second, these are positive sense single-stranded RNA viruses of zoonotic origin. Coming to the shape, they are spherical to pleomorphic particles and their length is between 80 to 160 nanometers. Next, COVID-19, it has 4 structural proteins. As you can see in the picture, it has the 4 structural proteins, namely envelope, second, spike glycoprotein, third is the membrane protein, and then comes the nucleocapsid. This is a picture as you have seen in the previous slide also. Next, these proteins, that is the spike, the membrane and the envelope proteins, they together form the envelope of the virus. In the figure you can see it clearly that the envelope, it is made up of 3 proteins, that is the spike, membrane and envelope proteins. Next, membrane protein, it is the most abundant and responsible for the shape of the envelope. And envelope protein, this is the smallest structural protein in this virus. Coming to the spike and the membrane proteins, these together they are known as the transmembrane proteins that are involved in virus assembly during replication. These 2 proteins, they play the role in the replication of the virus. Next, nucleocapsid proteins, they remain associated with the RNA of the virus, forming a nucleocapsid inside the envelope. The role of the nucleocapsid protein is that it is involved in viral genome processing, and it is also involved in other aspects of the COVID-19 replication cycle, that is, in its assembly and budding. The third function of the nucleocapsid protein is that it helps in host cellular response to COVID-19 viral infection. Polymers of spike glycoproteins remain surrounded in the envelope, giving it a crown-like appearance. Due to this, the name of the virus is coronavirus. This is spike glycoprotein details, and this is the conformation of the spike glycoprotein. The figure towards your right side explains the conformation of the spike glycoprotein. Now, spike glycoproteins, they have the two subunits, that is the S1 and the S2. Coming up to the S1 subunit, that is S1 subunit, it contains signal peptide, that is followed by an N-terminal domain, and the third, it has the receptor binding domain. S2 subunit, it contains conserved fusion peptide, heptad repeat 1 and 2, a transmembrane domain, and it contains a cytoplasmic domain. So, this is related to the spike glycoprotein. It has two subunits, and the detailed information about the subunit 1 and the subunit 2 is given. Next, the subunit 2 of COVID-19, it's highly conserved and shares 99% identity with those of the two bat SARS-like coronavirus and human SARS coronavirus. So, it is the subunit 2, S2 of COVID-19 that shares the similarity with the bat SARS coronavirus and the human SARS coronavirus. Next, now you'll get this idea from the picture, that is the spike glycoprotein, it binds to the human ACE2 receptor. From the picture it is clear that the spike glycoprotein, it binds to the receptor in the humans, that is ACE2 receptor. This receptor, it is present in the target cells in the respiratory tract. This protein, this spike glycoprotein, has a compact ridge that allows the virus to attach more strongly. So, this is the main advantage for the spike glycoprotein. It attaches very strongly to this receptor as compared to other viruses of the same origin, and that is why, as you can see, first of all it has a closed conformation, and then after binding it changes to an open conformation. Now, after the spike protein binds with the receptor in the target cell, the viral envelope fuses with the cell membrane. First the spike glycoprotein, it identifies the receptor, it binds to it, then the viral envelope, it will fuse with the cell membrane and releases the viral genome into the target cell. The target cell here we are talking about is the human cell, that is in the respiratory tract of the humans.
Friends, thank you so much for watching this video. In this video you saw that the molecular structure of the novel coronavirus, and this is the part 1 video. Part 2 video will be coming soon, and in the part 2 video we will discuss about the detailed molecular genomic structure of the novel coronavirus.